In this video we're going to discuss two different philosophies for discovering drugs. One is called target-based drug discovery. And we discussed this a little bit already. That is target-based drug discovery, oftentimes just written TBDD. And then the other philosophy is phenotype-based drug discovery. So target-based drug discovery begins with a drug target. This is a protein that is crucial in a biochemical pathway, and has been identified because that protein will give control over the pathway. So if you can bind a drug to that protein, then you will interfere with the pathway and hopefully affect the disease state. So the target is a protein. And once you identify this protein, you develop a binding assay, a means of measuring how a drug binds to the protein. So we need a binding assay. And this is in vitro test. It's something you can run. They're not run typically in petri dishes, but in small vials or small microtiter plates. So this is where we begin with a target-based drug discovery. And then we submit molecules to this binding assay, and we find out how well each compound binds to the target protein. And because of the simplicity of this binding assay, we test millions of molecules through this assay. So it's very easy to make new molecules and subsequently submit them to this assay, and get almost immediate feedback on whether your changes improve binding or are detrimental to binding. And so as a result, once you have your hits and you select leads, your lead optimization process is fast. Once you find something that binds, it's fairly a simple process to begin improving activity. That's not trivial, but it's a quick process. Well, this sounds wonderful. What is a disadvantage for target-based drug discovery? A disadvantage for target-based drug discovery, it all hinges on the idea that the protein that you selected will in fact have a significant biological effect once you bind to that protein. So what you're really optimizing through the study is binding. But if the protein is not effective, the molecule won't be effective. So the downside is sometimes you can develop a tight binding molecule, but the efficacy of that molecule will be low. And remember, in the drug discovery process, you don't learn whether your molecule is effective in humans until you get into phase 2 clinical trials. So it's been a long road, using a lot of resources. And to learn in phase 2 that your drug has failed. That's very hard news to take. As it turns out, many drugs indeed do fail in phase 2 clinical trials, because they lack sufficient efficacy. So this is a weakness of target-based drug discovery. So what's a potential solution to this? Well, many people advocate for phenotype-based drugs discovery. Well, what is a phenotype? A phenotype is any observable characteristic in an organism. It could be blood pressure, high cholesterol levels, sleeping patterns. It could be reflexes, anything that you can measure on an organism in that way is called the phenotype. And drugs can change phenotype. Drugs can change your blood pressure or your cholesterol levels. So these drugs change observable characteristics in organisms. It doesn't just have to be a living organism, a whole animal. It can be tissue samples. So you could look at how chemicals can cause muscle fibers in a culture plate to twitch or to contract. In fact, you can measure the strength of that contraction by putting tension on the muscle fibers. You can even look at phenotype changes in cellular-based systems, cancer cells. How quickly do they divide? How slowly do they divide? You're looking at observable characteristics of that cell culture. Are the cells alive or dead? That's another kind of drastic example of phenotype. But phenotypes focus on observable characteristics. And normally a phenotype-based drug discovery program will begin with knowledge about a molecule that has an effect on a phenotype. So, for example, I mentioned sleeping patterns earlier. Let's say you, as a drug company CEO, are aware of the fact that people in a certain part of the world, when they chew on a certain type of leaf, they become drowsy. And you recognize that, wow, those leaves, if they make people drowsy, that could be a cure for insomnia. And a lot of people suffer from insomnia. So you might send a team of people to go harvest some leaves, bring them back. We'll crush them up and we'll try to extract the molecules out of those leaves and identify which molecule affects the phenotype of people, their sleeping behavior. And you might find out what that molecule is. That will be your lead. So your lead has efficacy, and that is important. So what's the next step? Well, we need to then test our molecule, and our assay is probably not going to be in people. But we need some sort of experiment that we can perform with our lead that's going to be in some type of living organism. So this will be a in vivo tests or a cell-based test. That will give us insight into whether this lead has the effect that we want, causes drowsiness, whatever we're happening to look at for our particular disease. Well, as it turns out, in vivo testing, or cell-based testing, is not nearly as quick as a simple in vitro biochemical testing screen. So that's a problem. It slows down your lead optimization process. You could make new molecules, but testing those molecules in these living organisms is a much messy process. And it takes a lot longer to get your data back, so your lead optimization is slow. That is a problem. Time is money. And if you're spending a lot of time trying to optimize these compounds and you're doing it kind of blindly, that is a challenge to phenotype-based drug discovery. Of course, the advantage to phenotype-based drug discovery is that you're fairly certain that you're going to end up with a compound that has efficacy, because you know from the get-go that this molecule was effective in living organisms. So these are two different approaches to drug discovery. And I'd say target-based drug discovery, as molecular biology has advanced, has sort of become the predominant method. But some people, because of efficacy problems that they're observing in molecules that are advanced to the clinic, they're saying that we need to look back to what the drug industry has done historically, the phenotype-based drug discovery. Of these two philosophies, we're going to focus on target-based drug discovery. But if you continue studying medicinal chemistry, you will definitely encounter people who are strong advocates, for some valid reasons, for phenotype-based drug discovery. And it's important that you are aware that there are these two different philosophies.